welcome back to Geneducation. This is Juliet, my name is Larissa, and today, as you can see, we're in a little bit different setting. Today we are going to talk about PCR, Polymerase Chain reaction. And we are also going to introduce you to a team Leiden, that works with PCR. The polymerase chain reaction can be used to detect the presence of a gene or to amplify the genetic material for further use. PCR enables one to very rapidly multiply even the smallest amounts of genetic material, DNA or RNA, from a variety of sample types. As a consequence, this highly versatile and largely automated technique is appreciated in clinical settings and beyond.
PCR allows one to detect the presence of a specific genetic sequence and amplify it. When conducting a PCR, one has to find a sequence that is specific for a certain gene. As this technique is often used on samples from humans, animals, plants, or bacterial cultures, genetic material from many other organisms or contamination may be present. However, if the chosen region is specific in a single organism, accurate detection will be possible.
PCR is used for the diagnosis of many infectious diseases. Pathogens, bacteria, viruses, and parasites containing DNA or RNA. Some pathogens can hide inside the cells and as a consequence cannot always be detected using techniques such as antigen-based serology or microscopy. This is sometimes the case for malaria. Moreover, certain other techniques detect the reaction of the body to the presence of the pathogen, rather than the pathogen itself. However, these antibodies can sometimes only develop after a few days to weeks. In hospital settings, it can then be preferred to take a patient's sample and run a PCR test, using primers specific for a certain disease, such as malaria, SARS, viruses, or Ebola, for instance. In the case of the current pandemic, nasal swabs can be taken from suspected patients and run in the labs all over the world.
Yearly, many outbreaks of infectious diseases occur all over the world, such as malaria, cholera, and Ebola, impacting many lives. These outbreaks can be caused by viruses, bacteria, or parasites that infiltrate a human population in the course of an outbreak. Often, the outbreak can successfully be managed through the intensified personal hygiene, treatment, vaccination, or other additional measures. However, sometimes outbreaks can get out of hand and develop into epidemics or pandemics.
Our planet is also undergoing rapid and drastic changes: deforestation, rising water levels, intensified agriculture, and migration and population movements. Next to the already existing threat of normal pathogens, humanity is exposing itself to increasing occurrences of outbreaks of infectious diseases. This is often the case for pathogens that are zoonotic, which is the transmission of a pathogen from the animal population to the human population.
The changing climate can also affect the transmission of pathogens that are vector-borne, for example mosquitoes migrating towards the north, or waterborne, like rising water levels can bring infectious agents closer to humans. These all have the potential to pose a major global public health challenge. Team Leiden is on the forefront of fighting these diseases with their new diagnostic PCR method.
For years now, scientists have been warning governments about the very real threats that an epidemic, or worse, a pandemic, would pose to the world. The unknown upcoming disease is often referred to as Disease X. Despite ongoing efforts of surveillance, a lot of aspects are still unknown about these threats, or the nature of the pathogen. But the recent outbreak of SARS-CoV-2 virus have provided us with an insight of what we might expect. It also served as a true eye-opener for government leaders who realized they were very ill-prepared for the management of the spread of this pathogen and the capacity to test and prevent such an outbreak. In the future, tools will be needed to rapidly identify these diseases and prevent their further spread in this highly globalized world where one can travel half the world in a day. We need to take lessons from past and current situations to establish better collaborative networks for future epidemics and ensure a rapid and coordinated governmental response for better diagnosis. And this is why we've decided to develop Rapidemic. Rapidemic is a rapid diagnostic test capable of rapidly diagnosing infectious diseases. The kit is a point-of-care device that contains a simple and elegant 3-step reaction that is easy to use and will be able to successfully tell whether a patient is infected with a particular disease, as indicated by a color change. When an outbreak occurs, researchers first look at the genome of the pathogen, attempting to find a sequence or a part of this genome that will be able to conclusively identify all of the patients that bear or are infected with this pathogen. Researchers can then make this part that can recognize and bind the sequence of the pathogen. Only the very first step of the system is specific for the pathogen. Therefore, the significance of the application lies in its versatility. Rapid diagnostic kits could be sent out and stored worldwide in vast quantities. In the case of an outbreak, researchers immediately look for the specific part. This part is then added to the generic detection kits to form a complete kit. As a result, the kits can be used rapidly and specifically for the pathogen at the source of the outbreak. It is no longer necessary to make a whole new kit, but just add a small part to complete already available kits. Diagnosis is the first step towards treatment and recovery. Many infectious diseases caused by bacteria, viruses, or parasites require rapid and accurate diagnosis. Rapid nucleic acid testing therefore has the potential to reach lab-grade sensitivity and accuracy, directly targeting the pathogenic agent, and would not require extensive equipment. These kits have the capacity to be sent out globally for local use. The collaborative efforts can benefit the global population in quickly diagnosing novel emerging infectious diseases and tackling these at their source. In the process of development, our team has been thinking thoroughly about the applications, the setting in which these kits will be used, up to the hardware and the design of the kits. This is why we have contacted researchers, doctors, patients, NGOs, regulatory bodies, and business stakeholders in the diagnostic field. They are continuously helping us in the process of identifying patients' needs and how to optimally address these.